And I guess that's a good reminder. We will, once the meeting is done with, we will get that recording up on the YouTube channel, and we can put that link in the chat too that has, recent and all the previous meetings once we started the recording process. And so that's an easier way other than trying to have the presentations posted. So we do that. And so if you know of anybody that's interested or couldn't make the meeting, or you decide you want to go back, those are available. And thanks to Nick Moderano of the Water Quality Monitoring Council is always great at helping us get those posted. So thanks to Nick for that. Any other quick announcements? I guess I'll go ahead and let folks know I'll be stepping down as CCHAB co-chair, and so we will go ahead and have nominations open for those who are interested. You can self-nominate, or someone else can nominate an individual that is interested and willing to do so. And I think that'll be, I think, open for a month or so. We'll get an email out through the listserv with more details. So it's been a pleasure, and thank you all for being such a great group, and I look forward to having some other state or federal government representative to assist with taking this forward. So thank you.
Yeah, we'll be sad to be losing you, Becky. But yes, you said we'll be accepting nominations. But thank you, Becky, so much for all your service on this network. It's been really great to work with you, and I think you've done a lot for the community. So I think we all collectively say thank you for your service on this network.
No problem. Thank you. Well, if we don't have anything else, are we okay going ahead and starting a little bit early with Dr. Haney? If that works for you.
Sounds good.
Great. Are you prepared and okay sharing your screen directly?
Sure. Mm-hmm.
Okay.
Does that look all right?
That looks great. Thank you.
Okay.
So yeah, appreciate Dr. Haney being willing to speak today about that work on cyanotoxins in aerosols from Lake Tahoe, and appreciate your willingness. And without further ado, go ahead. Thank you.
Okay, thank you very much for the introduction, and also for inviting me to share with you some of the findings that we've gotten from our work on Lake Tahoe. Although it's very preliminary, it still raises some really interesting questions. Also, I wanted to mention I'm very excited to hear Haley Ploss today and about the work that she's doing with aerosols in a very contrasting situation. So I think it'll be fun to see both of these. So thanks for setting this up, and thanks for the CCHAB committee for all the work you're doing. I think this is really what it takes at the, if you think of it at the local level, at the state level at least, to have enough interest to investigate what is actually happening in the world of research and CCHABs. So thanks very much. And I'll begin by saying that what I'm going to share with you is essentially the report that we did. This was an amazing job of creating an investigation and concluding with a report within 4 months, the entire project was done. So I feel very proud because I usually don't get things done that quickly. So it was fun to see that you can actually do this if you hold yourself to essentially to the fire. So this is what I'm calling a preliminary investigation of cyanobacteria toxins in Lake Tahoe water and aerosols. This much of the work was done by Haley Carter, who's acknowledged here. She had just finished her master's degree about 2 days before we began this project. And she very generously was willing to continue on and essentially do the same type of work she had done for her master's degree, looking at aerosols. And in her case, she had done them on 2 lakes in Cape Cod area. So with that, I wanted to just say a few words about my background to give you sort of a context, and also the way we're looking at lakes and cyanobacteria. Well, first of all, my background is about a half-century of training students and doing research in freshwater ecology and ecotoxicology. I've just recently become professor emeritus, which sounds really quite good and has, like most things in life, trade-offs. I do less teaching now, and I have more time for research, and of course, a much smaller paycheck. So that's the trade-offs that we see. But it has released me to do much more in-depth work on this aerosol project, actually. So first of all, I wanted to mention to you, as far as my background, I first became involved with cyanobacteria in a project on New Zealand, where we were looking at lakes where the blooms would continue sometimes more than a year or a year and a half. And so we began looking at lakes that were very highly polluted. I came back to New Hampshire, which has about 650 lakes, the majority of which are oligomesotrophic lakes, so not very enriched. And so I asked the question, which of our New Hampshire lakes have toxic cyanobacteria?
have a lot of those meetings too.
And to answer the question, which lakes have toxic cyanobacteria, we had to develop sensitive methods for testing under these low productivity conditions. These gave me what I like to think of as essentially lessons from a healthy lake. And so the first lesson that we learned is that the toxic cyanobacteria were present not in just some of the lakes, but in all of the lakes of all trophic conditions, in the water, in the plankton, and in the sediments. So the idea that a lake is either toxic or non-toxic, at least where we've been working, is not really a reasonable question to ask. The second lesson from healthy lakes was that when we looked at our data, we discovered that in about 90% of the lakes, 80% of the microcystins, which is the toxin we were dealing with back in the year 2000, primarily, that the microcystins were in the smallest cyanobacteria, which we now refer to as picocyanobacteria. So that these were not the cyanobacteria that appear at the surface of lakes and pose problems that most people study as HABs. So this led us to think about the cyanobacteria in terms of their ecology. What role are they playing? And we have toxic cyanobacteria, but they're not fitting the simple eutrophic lake model, it seems. So when we looked at the transfer of the toxins, what are these toxins doing in the system? We found, of course, what other people had found is that the zooplankton that eat the cyanobacteria generally reject the large particles, such as, this is Vornicinia, that spherical one, see it's kicked out. And they also, other colonial types, such as Dolichospermum, the long stick-like cyanobacteria, simply don't fit into the filtering apparatus and they get rejected. What's going on now that you don't see is that very tiny picocyanobacteria too small to be seen with this light microscope are actually being consumed. So this led us to develop a relatively simplistic view of the toxigenic cyanobacteria in the plankton and the role that they have. And it's simplistic simply because it deals primarily with size. We have the large forms, the bloom-forming cyanobacteria, or BFCs as we affectionately call them. And these are larger than 50 microns, which is to say, generally they're not eaten by the zooplankton. And then we have the small other size realm, which is very small size, which is the picocyanobacteria, or PCYs. And these have a different role because the large BFCs are the ones that we often see at the surface. And these are the focus of much CCHAB research. They are very often highly toxic, a threat to recreation, drinking water, and wildlife often. But the PCYs enter the food web, and there they can bioaccumulate. A second role that they have is that they can enter the air because they're so small as aerosols. This is just a diagram that shows the same thing, where the large on the right-hand side are ending up as a surface scum here, a typical HAB, and what I call the edibles, the small PCYs, either go through the food web and end up in the top of the food chain, bioaccumulating along the way, or they will actually also reach the surface where they can become toxic aerosols, either the cells themselves or the toxins associated with it. So why are we interested in cyanobacteria in the first place? Well, one of the reasons, obviously, is they're a potential threat to public health. And I'd like to just talk very briefly about the 3 toxins that we are dealing with in this study that I'm going to be showing you. First is microcystins, the longest studied probably of the cyanobacteria toxins. And this is the one that we began studying in New Hampshire. It's very interesting because at that time it was referred to as a liver toxin, and it does accumulate in the liver and can cause liver diseases, but it also, we now know, has a host of other damage that it can do to vertebrates. The second toxin that we're looking at is a neurotoxin, anatoxin A, often referred to as the very fast death factor toxin. And it's the one most often associated with the death of wildlife and dogs and livestock, mainly because they often die near the source of the problem, which is the lake, and they die from drinking contaminated water. There's not much known about this toxin. In the past, it's been looked at as a very delicate toxin in terms of its half-life in nature. We've begun looking at it because we were quite interested in other neurotoxins that might be present in the water and in the air. As it turns out, it is not as difficult to work with as we once thought, although it does have problems with decay. And finally, then, the last toxin we are dealing with here is beta-methyl amino L-alanine, which is BMAA. And it's a very interesting, tiny little amino acid that doesn't really get involved in the building of amino acids used for proteins. It's been suggested as a possible environmental trigger for neurological disorders such as ALS and Alzheimer's through both field and lab studies. So I'd like to introduce you to a colleague from New Hampshire, is Dr. Elijah Stommel. And in some ways, he generated much of the interest that I have in aerosols by publishing a paper in 2013 on aerosolization as a potential risk factor for amyotrophic lateral sclerosis. And what he was doing is trying to explain why there are clusters of ALS cases, and he works at the ALS clinic at the Dartmouth-Hitchcock Medical Center in Hanover. And he found that in some cases, at least, there are clusters of ALS around certain lakes. And so he was conferring with me. We didn't know one another at the time, simply asking what's wrong with these lakes? Are there cyanobacteria in these lakes that could be a contributing factor? And so that got us started looking at aerosols and trying to develop simple methods with which we could collect and detect aerosols. This is Kate Langley collecting aerosols on Lake Mascoma, one of the lakes associated with a cluster of ALS patients that had about 24 times that which would be expected from the case distributions. So Kate did her first study, which was the major study on lakes in New Hampshire using this method, which was a study looking at 8 lakes selected to represent a wide range of trophic conditions. And she was using this what we call the CLAM, the Compact Lake Aerosol Monitor. You can see it set up here on a lake. This is about 6 o'clock in the morning as she was retrieving her samples from the night before. And basically it pulls it pulls air from these funnels, which have a screen on them, protecting them from lateral wind action. So you're trying to collect the samples close to the surface of the lake, draws it through a filter, which then collects the cells and toxins on the filter, and then it goes into a pump, which is in the box. Newer version of this, which we developed in 2019, we call the PD CLAM, and this is the one used on Lake Tahoe because we added traps to it that collect dissolved toxins as well. Things that go through, the air that goes through this filter then gets essentially collected in a trap. So what did she find? Well, very interesting. What she found is that doing these 8-hour collections during the day and at night on each of these lakes, she found, first of all, that the toxic aerosols came from all of the study lakes. It didn't have to be a eutrophic lake. And that it came both in the day and the night. And she was unable to show any relationship with wind, so that aerosols were not dependent upon the wind condition. If anything, wind seemed to decrease the aerosols, probably by adding lateral movement of air from the terrestrial environment. So here you can see the results she has, dark being the night, light being the day. And so that said that this is a very interesting process of aerosolization, non-wind generated. So what regulates them? Well, let's take a look at aerosols. One way to look at aerosols is from the work that's been done, is that there is a type of aerosol referred to sometimes as wind-generated aerosols or turbulence-generated aerosols. This movie above here that I think you should be seeing is showing aerosols at a dam, which is about 2 blocks from my house. And the water coming over the dam then generates conspicuous aerosols, you can see with a light that is shining on the dam. And so this is definitely a type of aerosol that actually is very common when you have big storm events, large lakes and marine systems. It's the most studied type of aerosol production. And it results in large particulates being taken from the water, as well as dissolved matter ending up in the aerosols. In contrast to what we've been looking at, which is what I call non-wind generated aerosols, they're linked to small scale, and I put this in quotes, evaporative processes. It's in quotes because I don't really know how it works. We do know that it occurs continuously, sort of 24/7. This is going on. both day and night, sometimes higher at night than during the day. It releases small particles, cells that are generally less than 5 microns, and dissolve molecules into the air, such as cyanotoxins and a host of other molecules that we're just beginning to study. So to try to investigate what are the mechanisms behind this non-wind generated process, we've been testing a whole range of physical, chemical, and biological factors with, I've got 3 teams of students in the lab that are doing projects on these, and we're beginning to get some ideas, but it's not a simple process. So this sort of evaporative aerosolization, we're trying to develop a model that would predict under what conditions do we find high aerosol production or low aerosol production. And we're not there yet. Out of Kate Langley's master's thesis work, an interesting graph came out of this. And this is, if you look at the amount of microcystins which is a toxin, in the water, and compare that to the percent of those microcystins which end up in the air, what you find is an inverse relationship. And so this was really kind of troubling, but also very interesting, because when we compared the very oligotrophic lakes that had low microcystins in them with the more eutrophic lakes that had higher amounts of microcystins, what we found is, in fact, the low level lakes had, at least in her study, at least 30 times the aerosol rate release that the more eutrophic lakes had. So this is what I talk about as the aerosol enigma of healthy lakes. In other words, why is it that these clear, low productivity lakes can release relatively more toxins into the air than the conspicuously polluted lakes? And when you think of clear, low productivity lakes, of course, we think of Lake Tahoe. So I want to just pull up here, Lake Tahoe. So this is what I call the first assessment. You know, with that background, you can see that we were very interested in what happens with cyanobacteria in clean lakes. Are they present? What are they doing? And for us, the opportunity to look at Lake Tahoe was sort of like looking at the Galapagos Islands for a biologist, because we hoped that we would see some things happening there that would give us clues as to how this works. So let me talk about the project that we did. First of all, it was clearly a citizen-based project or citizen-initiated project. We were contacted because of citizen concerns with what was perceived as a high incidence of neurological disorders, such as ALS, in lake residents. There also were some developing water quality issues, especially in a development region in the south part of the lake, known as the Tahoe Keys development, as well as in the lake as a whole, which has then increasing production going on in the phytoplankton, and especially in the picoplankton. So things were happening there. So in talking with the citizens, we came up with some very simple questions that could be done on a very low budget. In other words, we didn't have time to develop a grant application here and wait for this. This was done very, very quickly, and we said, well, here's the minimum we could do this on. Here's the budget. We could at least get our feet in the water, so to speak, and see what's going on in Lake Tahoe with relative to cyanotoxins and aerosols. So, and the other is we at least would look at the toxins that would allow us to consider potential health risks that might be involved. So the target toxins were then the microcystins, the anatoxin, the neurotoxin, and the BMA and neurotoxin. Then we had a sort of a liver toxin, general toxin, and two neurotoxins that are very different in how they affect organisms, especially vertebrates. The anatoxin A is a fast-acting toxin. The BMA appears to be a much more slow-acting toxin. So let me just talk about what our goals were and where we decided to do our sampling. First of all, the Lake Tahoe has been manipulated in a way that provides sort of a natural experiment. In other words, the South Tahoe Keys isolated a section of the wetlands and water coming in, so that it now is surrounded by houses, and the water quality is conspicuously different from the rest of the lake. So we said, let's take a look then, as a starter, at the South Tahoe Keys, and then try to have a relatively nearby control site, which would reference site, let's call it, which was sort of representing the open water, even though it was only a few miles away. And this was, we found a location where we could run our samples easily from Valhalla Pier. So these were the areas where we collected water samples and aerosol samples. Hold on just a second here. There we go. Then we also had interest in the Serene Lakes area, which is in the northern section of Lake Tahoe region, in which on a small mountain area there are a couple of lakes associated with a small community where, again, it was perceived that there was a high level of neurological issues going on. So we looked at the Serene Lakes, but we didn't have the time or the budget to actually do aerosols on them. So we just took water samples, or had grab water samples taken for us. Okay, so let's take a look at these two sites. This is the South Tahoe Keys development area, and Keys Site 1 is where we ran the aerosol collections. The Site 2, which is the Valhalla Pier, then represents our more open water area. And we did two dates, one in July, where Haley Carter actually flew out to Tahoe and brought along the clam and ran the clam at these two sites, on-site aerosol collections, and then froze the samples, brought them back, and processed them in the lab. The August samples, we decided to do something we've done in the past with other lakes, which is actually process the water freshly collected in the lab, so we could control a lot of the external conditions, such as wind, elevation, a whole lot of things that can be controlled in the lab, but the water is being tested for its potential aerosols. So what we did then is August had an overnight shipment of water collected, and the next morning was received in our lab and processed immediately. So that worked out quite well, actually. We had the containers with all the water from the collections, then refrigerated and sent overnight. So when we received them, they were still at refrigeration conditions. So what did we find? Let's take a quick look at some of the results. First of all, start off with the simplest, which is if you look on the left-hand side here, or I'm sorry, the right-hand side for you, which is what I call the extra samples. These are the grab samples collected just to see, are these toxins present in the water? And this wasn't even quantitative. It wasn't done by going out into a boat and doing some sort of integrated water tube collections. This was just hand collections. And then we measured how much of the three toxins were present in each of these. And these were done at one of the extra Keys sites, not the primary site, two of the Key sites, and then at the Serene Lakes, what we call the main, Serene Creek, and then Serene Public Dock, and then a nasty looking drain, I believe. I wasn't there to collect it, but from the results, what we can see is that BMAA was present in all the samples, but at the storm drain, it was the highest in that collection. So if we look at the other serene lakes, which we were quite interested in, that we found that microcystins were either below detectable limits or found in only one of the sites in low quantities. So microcystins is not one of the big toxins we're dealing with at Tahoe when it comes to the water concentrations. We don't know how, of course, these were just water samples, what the aerosols would be like. And then the ATX, the same way, the ATX was quite low in most of the serene lake waters. So we suspect that BMAA would be something we should look at maybe further in the serene lakes, or maybe all three, just because we haven't got what I would consider to be a quantitative sample from there. So I want to very quickly then just go through some of the results of the primary sites, which is the Keys and the Valhalla Pier. So let me just show you what these look like. So first of all, if we look at the water and what we found in the water for microcystins, it's very easy to see that what we found, first of all, is that microcystins were low relative to a lot of other lakes, including New Hampshire lakes that we find. The concentrations were low in the Keys, which the Keys have very conspicuously green water, so that it's highly productive. And yet the microcystins were relatively low there. They were non-detectable, even using, I think we were doing a concentration of about 150 times for the lake water, and we still didn't detect it in the lake water at the pier on either occasion. So when we look at the aerosols on the right-hand side instead, what we see is a very different picture, in that the aerosols are present in the Keys, but in the at the pier site, the aerosols, even though we of microcystin, even though we didn't detect the microcystin in the water, it was so low, were present in the air, but especially in the August sample, were higher than we normally get, or we've gotten from eutrophic lakes that Haley Carter was studying. And I put in there as a reference. There's not enough studies of aerosols to do a statistical comparison. But what this says is, for the highest she got for Little Mill Pond in Massachusetts, the maximum level that she found was about 500 picograms of microcystins per meter squared, or meter cubed, sorry, of air, whereas the level that was found at the pier was much higher than that. So that was very interesting. One of the questions it raises then, are we talking about an extension of the aerosol enigma of healthy lakes that I showed you earlier? That's one of the questions. We don't know why, but is that what's going on here? When we looked at the very fast death factor, ATX, or anatoxin A, it was interesting that we found it in the water at both sites. On the August date, at least, we found it present in the purple column is in the small fraction. So in the open water, the toxins appear to be in what could be, well, the picocyanobacteria. And in the Keys, it's much more of a typical of eutrophic lakes, where a lot of the toxins are produced by the larger fraction, which is shown as WLW as the whole lake water. So that's unfiltered. So on the other hand, the aerosols are very interesting because there's almost no particulate aerosols with ATX at any of the sites, so that they're almost entirely dissolved ATX, which raises a question of what's happening here with these aerosols. A lot of the aerosols we see in lakes in the Northeast, at least, are caused by both the cells coming out of the water, having the toxins, as well as dissolved toxins being emitted. Here it looks like it's probably the dissolved toxins that are in the water, although we were unable to run dissolved toxins in the water with our modest budget, so that has to be a question for the next time. In other words, is the ATX that's present in Lake Tahoe coming from a dissolved fraction? So in the ATX that we saw at the Valhalla pier were 5 times that as the Keys, where you have a much more productive system. So what this is showing is something very, very unusual. The ATX, the antitoxin A, is not related directly to the productivity of the system. Okay, we're almost there. So let me show you the last one here, which is the BMAA, and one of the most interesting for many people because they're interested in, is there a connection with disease? And what we see is BMAA was widespread. We found it in the water. It was interesting that it was in both the particulate and the dissolved form, or not the dissolved, in the large particulate as well as in the pico fraction. The interesting thing about this was that the BMAA was highest in the Valhalla pier. And you can see it here on the August sample. It was above 150. Actually, that's just for the water traps. In other words, that's the dissolved. So it was not quite 200 picograms or nanograms per meter cubed of air. So it was quite high. And if we compare that to what we find in our eutrophic lake in Massachusetts, it was much higher than that. In fact, it was 3.5 times what we found in Little Mill Pond. So these are very high, relatively high aerosol rates. We don't know whether that means that people are having issues from that, but at least it's raising some interesting questions. Okay. So, what I wanted to show you is suggests also a possible link between BMAA and the picocyanobacteria, which should be looked at. In other words, picoplankton is the major producer in Lake Tahoe as a whole. And so the question is, are these toxic with BMAA? So next steps, if I can very briefly say what those are, we need to look at the picocyanobacteria in Lake Tahoe. They seem to be behind a lot of the aerosol we're seeing. And I'd like to do some genetics work. We're just starting this with our lakes work in New Hampshire, and ask whether or not they're toxigenic, and are they a species that we maybe is unique to Lake Tahoe in some way? Are they a species that's predisposed to becoming aerosolized? You know, when you think about it, getting into the air may not just be an accident. It may be a means of dispersal for some species. So the question is this unique to the Lake Tahoe area? So we would like to do some work on that. We need to investigate the serene lakes more thoroughly. There are toxins there. The question is, what are the aerosols like? And then I would simply point out, as you can already see, that this study was what I'd think of as a snapshot, both in time and location. And we need, if you want to understand the role of these toxins, we need to do a broader spatial and seasonal coverage of what's happening there. And also, it'd be nice to get more people involved in the health concerns human exposure data is needed. We need to collect data in a way that actually says this is how much you're being exposed to. In the report, I do some speculative calculations, which is if you were to breathe the same air that we're getting into our collectors, here's how much of the toxin you would take in. It's not necessarily trivial, so I think it's worth looking at, but there are a lot of assumptions being made there. And some of these assumptions involve things such as realizing by doing these 3 toxins here, you can see that when you breathe in the air, and we're breathing, about 1,000 to 10,000 times much more air into our bodies than we are drinking water. So small amounts can add up. And when you do that, the question is how many other toxins are you taking in? So what happens if you take in BMAA and then also anatoxin A, both neurotoxins? Is there some sort of interaction between those two? Are they additive? Is it synergistic? So these are some of the questions we'd like to get into. And it's what makes estimating human exposure difficult, is that there's a lot of other sources, such as how much you take in with your daily food when you eat a meal of these toxins. So I think to address the health concerns, we need a larger study group to participate. And finally, it would be very interesting with the focus on picocyanobacteria to see what's going on with the food web. And is there accumulation in the invertebrates, in the fish, and any of the top predators in the system? So with that, I thank you very much. And I want to thank Haley Carter. I don't know if she's here today looking at this, but she may be. She has been essential in this whole work, and we're still working on this. We want to try to come out with a small publication of this. Finally, I want to thank the grassroots support, and I don't think we've ever done a project with more grassrootsiness than this. And it was initiated by Trish from Tahoe area, and Trish has essentially been the driving force behind much of what we've done. And she's managed to make this project happen, collection of money, organization, and all those things, and then a host of other people who have contributed either time and or money to this project. And it has taken us a long way for a very small price. So thank you very much. I'd be happy to answer any questions if there's time.
Thank you. If you're willing to put your email in the chat, I know there's one question in there, but we need to move on to the next presentation to stay on time. So thank you very much for the presentation. And if you're willing to put your email or follow up, I know there is one in the chat now, or vice versa. Jen, if you're willing to put your email directly to Dr. Haney, that would be great too, whichever you prefer. Thank you. Okay. Well, without further ado, I want to welcome Haley Ploss to talk about similar work on cyanotoxins and aerosols from the Delta, and you should be able to share your screen.
Hi, thank you so much.
Thank you.
My computer just informed me that it's not going to turn on my camera today. I'm going to leave that off, but I will go ahead and get started. So exciting to see all of that work, Dr. Haney. I guess my presentation is going to be, I guess, a little less about cells that the cyanohabs can emit, or rather, I guess not the harmful algal blooms themselves, but also the picocyanobacteria getting into the air. But I'm going to talk more broadly about some of the metabolites, other than just cyanotoxins, that we're looking for as emissions from cyanobacteria, and specifically blooms of toxic cyanobacteria. So thank you all so much for having me today. My name is Haley Ploss. I'm a PhD candidate at UNC Chapel Hill. I'm advised by Hans Pearl, and I'm really excited to share also very preliminary data from our work in the Bay Delta. So as you all are likely aware, work over the past couple decades has demonstrated that cyanotoxins can go airborne, but we still don't know a whole lot about the environmental drivers of these toxins getting into the air, the cells getting into the air, and also the other metabolites that can be aerosolized. And as Dr. Haney mentioned, we don't know a lot about what the human health risks surrounding the inhalation of these toxins in cells looks like. So my work is also trying to better understand the environmental drivers to better inform public risk assessments regarding the blooms and their aerosol emissions, primarily in residential and recreational water resources across North America and beyond. So I exist in that measurement space as well, out in the field, trying to better characterize these aerosols that are emitted and the environmental conditions that promote their release during bloom conditions. So a quick overview of what we currently know about cyanotoxins and their metabolites in air. 3 cyanotoxins have been quantified in aerosol to date, and this is in the airshed of recreational water resources. So that's microcystins, nodularins, anatoxins. And one study specifically found that more hydrophobic forms of microcystin, such as microcystin leucine alanine, are more likely to be enriched in aerosol. And this is because of the hydrophobic properties of those side chains. So leucine and alanine is more likely to go airborne than, say, leucine and arginine for MCLR. People have also detected DNA of various harmful cyanobacteria in aerosol that is small enough to be inhaled, suggesting that some of these cells from toxic genera, such as microcystis, or fragments of these cells or some of their proteins are also getting into aerosol that's small enough to inhale. More recently, lipopolysaccharides from cyanohabs have been detected in aerosol. More generally, over the Great Lakes, it's been found that more bloom activity in the water itself increases signals of organic matter in lake spray aerosols. And then something that I'm going to focus on that we've seen in some of my work in North Carolina is that concentrations of particulate matter or aerosol in the ambient atmosphere have been found to increase in association with toxic cyanobacteria in the water column. So focusing on that last bit there, this link between blooms and decreased air quality or increased particulate matter in the air. Back in 2020, my lab group and I performed a field campaign in Eastern North Carolina, which is very similar to the work that I will talk about that we are doing in the Delta. So what we did is we looked for microcystins and cyanobacterial DNA in the water column and also in PM 2.5 or particulate matter with 2.5 microns in diameter and lesser. And this is in the airshed of the Chowan River in Albemarle Sound in North Carolina. So we had a pretty low toxin producing year. We didn't quantify very many toxins, microcystins specifically in our water samples. And we also did not quantify any microcystins in our aerosol samples during this campaign. But what was really interesting about this study is that we found a link between the abundances of dolichospermum in the water column and ambient concentrations of PM 2.5. So PM 2.5 is dangerous when inhaled, regardless of its composition, because it's small enough that it can penetrate deep into the human lung and possibly cross into the blood barrier. So this is a criteria pollutant of the EPA, and no matter what this aerosol is comprised of, it's not good to be inhaled. It's linked to cardiovascular and respiratory mortalities and morbidities. So what we found is that when dolichospermum increased, air quality got worse. However, this wasn't attributed to the emissions of those toxins that we looked for, nor was it attributed to increases of dolichospermum in our aerosol samples. So we think it was something else coming off of this bloom that was associated with this increase in aerosol concentrations. And what we hypothesize is that this increase could have been due to aerosolized metabolites other than microcystins. And what we thought might be a potential option was secondary aerosol formation from geosmin and 2-methyl-isoborneol, which are 2 common taste and odor compounds. So if you are unfamiliar with the processes that lead to secondary aerosol formation, this occurs when something is emitted to the atmosphere in the gas phase, and then it gets processed by other atmospheric pollutants and oxidants, and it becomes a lower volatility compound, which then condenses into a liquid or solid form, forming that aerosol phase compound. So it starts off as a gas, but it ends up as an aerosol in the atmosphere. And so we know that Plants emit VOCs. So trees emit isoprene, which is the largest source of secondary aerosol to the atmosphere. And we also know that marine phytoplankton are known sources of these gases, which contribute to secondary aerosol formation over the open ocean. But this phenomenon has never been studied in the freshwater for cyanohabs or other freshwater algae. So we were interested in understanding if geosmin or MIB as taste and odor compounds emitted by these HABs could be potential sources of aerosol. And I promise all of this background is relevant for our fieldwork in California, but I'm going to go a little bit into some of my other dissertation work, which is experimentally generating aerosol using geosmin and 2-MIB. So using a small atmospheric simulation chamber known as an oxidative flow reactor, we injected standards of both geosmin and 2-MIB in separate experiments into this chamber to react with hydroxyl radical, which is a ubiquitous daytime atmospheric oxidant. It's found everywhere. It's produced from ozone and sunlight. So when it reacted with hydroxyl radical, we measured the real-time aerosol production from geosmin and MIB. So we looked at their particle size distributions, what size they were, because that has, it's relevant to human health. And then we also measured how much of it was produced. And then we collected that aerosol onto a filter for chemical characterization using non-targeted liquid chromatography coupled with tandem mass spec. And what we found is that in the lab, both geosmin and 2-MIB yielded a lot of aerosol above our background measurements. Ultimately, geosmin was better at forming aerosol when we injected 100 parts per billion of the volatile compound. 33% of it was converted to aerosol, forming up to 230 micrograms per cubic meter of aerosol mass, which is like smog level or smoke level concentrations of particles. So that's something you couldn't even see through. And then when we injected MIB, around 15% of this was converted to aerosol, forming up to 100 micrograms per cubic meter of aerosol mass. And it's very important to note that in the bottom two graphs you see here, these are the size distributions. This is showing that this secondary aerosol that was produced is in the ultra-fine size fraction of aerosol, so less than 100 nanometers, meaning that this aerosol is very, very small, and it's small enough to penetrate deep into the human lung when inhaled. So this is the aerosol that you don't want to be breathing because it's bad for human health. And when we chemically characterize this aerosol, we found 8 distinct chemical features from our MIB-derived aerosol and 10 distinct features from geosmin. And then from these masses and the mass spectra data, we were able to generate proposed formulas and chemical compositions for this aerosol. And then we were able to take those masses and look for them in our field samples from California. And any detection of this would be indicative of cyanohab-derived secondary aerosol in the field. So now that I've run through a very quick crash course of what we've been looking for as far as aerosol emissions coming off of HABs and what is currently known, I'll get into the specifics of our work in the Bay Delta. So broadly, we're measuring aerosol emissions during harmful cyanobacterial blooms at 2 sites on the Delta, including Discovery Bay and the Stockton waterfront, since these are residential areas where people are more likely exposed. So we're trying to better understand health risk and the distance that aerosol produced might be traveling over land. So first, we're quantifying 3 different cyanotoxins, microcystin, anatoxin, and that should say cylindrospermopsin, not nodularin. We're quantifying them in both PM2.5 and PM10 because we're also very interested in the whether these toxins are transported in the cell or in the dissolved form, as Dr. Haney mentioned. And also, of course, if we were to see higher enrichment in PM2.5, which is smaller, this would be worse for human health. So that size fractionation is important for the risk assessment associated with our study. Secondly, we're measuring DNA in aerosol and generating metagenomes from some of these specific samples. So we're interested in any insight on genetic factors that might be driving the aerosolization of cells. Thirdly, we're looking for those secondary aerosol tracers that I mentioned that we produced in our lab experiments. And then lastly, we're generating water data, scraping the web for weather data and ambient concentrations of aerosol to put all of these findings into perspective. So to achieve this, we installed these very large, very high volume samplers at both Discovery Bay and in Stockton. So in contrast from Dr. Haney's work on Lake Tahoe, these samplers run for about 8 hours a day for an entire week at around 1,000 liters per minute. So we're collecting a huge volume of air onto each of our filters and integrating those samples over a week. So these run during the daytime from 9 a.m. to 4 p.m. And then these filters are being analyzed for the toxins and the SOA or secondary aerosol tracers that I mentioned. And when we collect each of these aerosol samples, we also collect a water sample for comparison to what we're seeing in the air. And then to generate the genomic information that I mentioned, we're using a lower flow approach. So I think we actually used the same pump that Dr. Haney showed, but we're collecting our DNA samples onto a filter and also via an impinger, which samples directly into a liquid medium so that we can analyze the cells that we're collecting in aerosol via flow cytometry. And then we hope to generate metagenomes from our filter samples. So as I mentioned, to put all of this into context, we're also generating information about what's going on with the blooms in the water. So we're looking for cyanotoxin concentrations, chlorophyll A concentrations, nutrient concentrations, and DNA or diagnostic algal pigments in the water to get an idea of what the cyanohab activity looks like. And we're also borrowing geosmin and MIB data from the Department of Water Quality. So the site that they have closest to our Discovery Bay site is the Clifton Court 4 Bay. So we're looking at geosmin and MIB data from Clifton Court in comparison to what we're measuring in aerosol. And then we've yet to analyze any of our weather data or ambient air quality data, but this will be scraped from nearby National Weather Service stations, and our ambient particulate matter data will be scraped from the Purple Air Sensor Network for nearby sites. So our work is also very preliminary enough that I actually just received the cyanotoxin data on Monday of this week. But we did not quantify any microcystins in any of our PM10 or PM2.5 samples collected from Discovery Bay and Stockton in 2022. And we don't think that this is due to our limits of detection for any reason, because we using liquid chromatography with mass spec, it was very sensitive. So the highest maximum concentration of microcystin that could have been observed in aerosol that we would have missed based on detection limits would have been somewhere around 30 to 250 femtograms per cubic meter. So very, very low concentrations. It's a very sensitive method. So it's possible that microcystin just wasn't getting into the air, or the high volume approach doesn't work. This is something that we're discussing. We're discussing for this upcoming summer. But I do also want to mention that we did spike some filters with microcystin leucine alanine as control to make sure that our method was sound, and we did end up detecting the microcystin in our control samples that sat out in the field all week. So there were some quality assurances here. We feel fairly confident that we just weren't measuring particulate microcystin. And in theory, our method would have collected microcystin even in the dissolved phase as well. So we still don't have any information about our anatoxin or cylindrospermopsins. We're waiting on those results. And we also don't know what toxin level we were starting out with in the water. Those samples are all still being processed, so that this part of the story is to be continued, and we're rerunning these collections this summer. So although we didn't find any microcystins, we did detect 2 SOA tracers that were derived from MIB. So here we're showing a chromatogram and fragment ion spectra that shows the match between our experimentally generated compounds and what we detected in PM2.5 at the Discovery Bay site. So as you can see in the top panel, the compounds exhibited very similar retention times. In the bottom two panels, you can see that the diagnostic fragment ions are a match in exact mass and relative abundances. So this is what's suggestive that they are the same compounds, even though we don't have any sort of commercial standard, since this is the first time that we've looked at SOA produced from MIB or geosmin. So this is very, very exciting to us because this would be the first evidence to suggest that secondary aerosol can be formed from volatile metabolites emitted from the cyanoHABs. And what's even more exciting about this data is that the MIB measurements at Clifton Court do circumstantially confirm these measurements. So all summer, low concentrations of MIB and geosmin were measured at Clifton Court, but in early August, the levels of MIB spiked at Clifton Court, and then 2 weeks later is when we started actually measuring these MIB tracers in our PM 2.5 samples. So we identified these tracers on 3 different samples that were collected between mid-August and late September at Discovery Bay. And we think it's possible that we were measuring the SOA that was initially emitted from Clifton Court, and then we were measuring it downstream at Discovery Bay, or it's possible that the blooms in Discovery Bay were also generating geosmin or 2-MIB, even though we don't have those discrete measurements. So we're hoping to use this bloom data and air mass movement data that we will collect soon to explore this more and put this more into context. So this work is nearly ready for submission. We're hoping to publish this later this summer. And then I wanted to end on what we currently know about ways that you can protect yourself. So although we do not know very much about the toxicological effects from inhaling cyanotoxins or metabolites coming off of the HABs otherwise, what we do know is that highly efficient masks and HVAC filters can block aerosols that carry microcystins. So a study from the University of Miami a couple of years back measured the efficiency of masks to block aerosols specifically carrying microcystins. And the good news is that in the field, if you were wearing an N95 mask, it would block an aerosol carrying any of these metabolites emitted from the blooms, just like it would block an aerosol that would be carrying COVID-19 or a respiratory pathogen. So in summary, we didn't quantify any microcystin in our aerosol from the Bay Delta to date, but we're still waiting on other cyanotoxin data and in both the air and the water. However, we did demonstrate for the first time that SOA can be generated from taste and odor compounds, specifically MIB, and we measured this at Discovery Bay. So this poses the question of whether taste and odor compounds are truly non-toxic, since the inhalation of ultrafine aerosol is associated with negative health effects. And as such, our kind of big takeaway is that we need to think more broadly, other than just cyanotoxins, when it comes to the impacts that harmful cyanobacteria in the water can be having on air quality and human respiratory health. So that brings me to the end. Thank you all so much for having me. Here is my list of all the amazing researchers who have made this interdisciplinary study possible. And I have a reference list that I can share if anyone is interested, and I'll take questions at this time.
Apologies, we are also at time for the break. So if people have questions in the chat or you've got your email up there, which is great, but we do have a 10-minute break scheduled. So if people want to go ahead and take that, we'll reconvene at 10:15. Defer to you, Haley, if you want to take anything live during the break, or if you want to go ahead with things followed up by email or in the chat. So thanks everybody, but we will be starting with our next agenda item at 10:15. Thank you. Well, thanks everybody for coming back from the break. We're going to start doing our regional water body HAB monitoring report, and Carly's going to kick us off with a statewide FHAB program update, as well as I think covering some of the regional programs. We generally go north to south after that. So I know Mike mentioned he has slides. If other people are going to be presenting slides, if they can send Jamie a direct message so she can make you co-host. Otherwise, we'll kind of run through once we hear this. And then we'll run into the subcommittee and work group reports. So thanks, and welcome, Carly.
Yeah, thanks for having me. So this actually is going to be pretty quick for this quarterly meeting, just because thankfully we've had somewhat of a quiet winter on the FHAB front. So with that, I'll get started. Okay, so first, for those of you who aren't familiar, State Water Board FHAB program is comprised of 5 funded staff, including both Marissa Van Dyke and myself. And we are the FHAB Freshwater and Estuarine from Algal Bloom program managers at the State Board. And then we have 3 funded staff at regions 1, 5, and 6. You may notice the red for region 5. They're in the process of hiring a new FHAB coordinator for their region. So we're excited to onboard somebody before the beginning of the field season, hopefully. And also, just a reminder to everyone that we maintain the FHABS portal, which includes our map, which is updated in near real time of any HABs that are reported to us as an agency. We have a ton of resources on there for the public on how to stay safe. Please borrow, use, share at your discretion. We also have advisory frameworks, our FHAB response plan on how to respond to a bloom, and a lot of resources for agencies and other groups doing some monitoring on some standard operating procedures for collections and things of that nature. With any questions, you can always email us at that email below. And just kind of going over really quickly our response plan that's on our portal that is designed through the CCHAB network. We have a flow chart here. We actually have thresholds, and then as well are signs that we ask people to actually put up at those water bodies. And this is just to kind of give everyone a breadth of what blooms have been reported to us in the last couple of years. We always just like to highlight that the numbers keep going up, yet our staff volume numbers stay the same. And now to the fun stuff. So I just want to put in a big plug. We're doing our holiday assessments again this year. Most of you hopefully are familiar at this point. They started in 2018, so this will be our 6th year. If you are interested in participating and sharing your data with us so we can get it on our map before all the major holidays, please go ahead and reach out to us. Marissa, if you could put the link to the survey in the chat, that'd be awesome. And we will follow up with you. So some things that we request that you provide to us: the sampling location, as well as the testing results, the presence or absence of a visual bloom, and we can provide some materials to you, such as sampling kits and that as well, to help support that effort. So once again, we're asking that folks get some information to us by May 9th. So the deadline is coming up, so mark your calendars. Another cool update is we've been working with our contractors at SFEI, the San Francisco Estuary Institute. We maintain a satellite tool map that we use for screening for HABs, and right now it has the CI cyano on it, but we're going to be integrating in the chlorophyll A from the Sentinel-3 data. So that'll be a whole new addition. We're piloting it now, and we're hoping to get it on our webpage hopefully by the end of May, and we'll be sending out an email to the CCHAB network email subscription list. So keep your eyes out because it'll be a pretty cool tool. We're always open to some input. So once you start diving in, if you have some suggestions or other ways you'd like the data presented, don't hesitate to reach out to us. And then we are amping up for our 2023 field season. As I just mentioned, we're getting ready for our pre-holiday assessment for Memorial Day, where we'll do the monitoring starting in mid-May. We also have quite a few regional board special studies that are going to be started this year. I don't want to take all the credit for the regional boards, but a lot of folks aren't here today, so I might just give you guys a little taste of what some of them are working on. Region 9 is going to be looking at the Santa Margarita watershed. They had some detections of some benthics there in a previous study, so they're going to be taking a little bit of a deeper dive to really understand the compositions. They're also going to be looking at some of their reference streams. So they do bioassessment through part of a reference condition monitoring program. So they're going to add in collection for some algae for toxin testing, once again, benthic related. Region 1 is going to be looking at the benthic stuff again in the North Coast region, some of their major rivers. And we're also going to be implementing some partner monitoring programs. We're lucky to have quite a few this year. So Region 5 is going to be collaborating with Restore the Delta and with California State Parks up in the Gold District to do some monitoring. And Region 1 is going to be partnering with the tribe to look at Big Lagoon. Region 2 is going to be partnering with a nonprofit to look at the Bay Area. I think it's like Merritt. And then forgetting one. Oh, Region 6 is going to be partnering with Inyo County to be looking over at the Eastern Sierra, some of the water bodies over there. With that being the case, if you would like to join our partner monitoring program, we're always welcome more partners. Please feel free to reach out to us anytime. And then one new addition we have that we were really excited about is we developed a new wiki. So it has some training materials for how to do some monitoring, tutorial videos, visual monitoring tools, a whole host of things. And because it's so exciting, I'm going to give you guys a quick demo. So bear with me as I switch screens. Okay. So this is our new wiki, and I'm sure Marissa will put it in the chat as to the link, but it's available for everyone. And it's really focusing on that monitoring component. So if you want to join us with the holiday assessments or the partner monitoring program, this is a great place to start to get some information. So we have a link here that just gives you a little bit of background, FAQs as to what HABs are, and then a lot of links that actually direct you back to our portal to give you some of those resources. A link here to get to know our team and who might be the most appropriate regional board contact for your FHAB coordinator to get involved. It includes email, so it's a really easy spot to one-stop shop. Here we have our partner monitoring module, and this kind of gives you some ideas as to, when we request partner monitoring, how to pick what kind of monitoring or flyer. And then down here is kind of the really cool component that's pretty specialized to this wiki. So if you want to learn more about visual monitoring, you can click on this link. You can also click from over here. And this will tell you a little bit on when to visually monitor, a link to the ITRC YouTube video, which is really awesome resource in terms of knowing what to identify, and then our visual guide. And then also field tests. So we have some information on a jar and stick and gloved hand test, who performs the tests. And then really cool, and the other thing is we actually have videos to actually guide you on how it would look to be doing some of the sampling. So I know it's like some people are visual learners, some people like to read it, but our FHAB coordinator staffs were awesome to video themselves doing some of these tests so you can see what they look like. And then we have field kit tests. So this is going to be some of those rapid test kits, including Abraxas. We have some resources on that and some training videos. If you're trying to get out there and do some lab sample monitoring, we have some materials on that as well. And we separated it both into planktonic and benthic. So if you're going to be going out and doing some planktonic water column sampling, we have in here our SOP, our visual guide, and then as well as some videos on how to do that grab, scum, and depth integrated sampling, as well as a monitoring plan template, supply list, and then we always have, in all of these, we can have the COCs and all of that additional information, so in our laboratory list, so you can have all that information. Benthic is very similar as well. And then we do have another section that we're working on that's specific to SPATs. So that's that continuous monitoring. And that includes how to actually build them. And we'll be getting some more videos and informational items on that into the future. So we're really hoping to build it out so that it's a good resource and you can have your one-stop spot to go and get all this information and pull from it. A lot of this, as I said, is also on the portal, but we wanted it to be a little bit more interactive, and having a wiki gave us that opportunity. And with that, I really do want to thank our FHAB coordinators because they did all the heavy lifting on this. They're the ones that train a lot of our partners, so they have a really great perspective on what would be needed. So big shout out to you all. And then once again, if you have any questions, comments, or concerns, you can always reach out to us in our CCHAB inbox. And with that, that's all I have.
Great, thanks, Carly. Did you want to cover any of the regions? Did you have anything from them that can attend? I know I got slides from Carrie.
The only update we received was from Region 2, and they just wanted to update about their partner monitoring program they're doing this summer, and that they have some interested other folks that they may be onboarding, as well as with the San Francisco Bay incident last summer with the red tide. They're really mobilizing to get a better plan together for if a situation like that happens in the future. They have a full plan in place so they can better address it. So they are taking a lot of precautions and doing a lot of planning this winter. And Marissa, jump in if you have any other updates from any of the other regional boards, but I think that's mostly what we got.
Yeah, I don't have anything to add from the regional board updates. But we can go through, from north to south as we normally do and see what other updates we have from other organizations. Everyone's welcome.
Great, thanks. I know Mike said he had slides for Region Board 1, so we can start up north like usual.
Okay.
I think display settings swap should.
Yeah. Awesome. Okay, good morning, everybody. My name is Mike Thomas. I'm the FHAB coordinator for the North Coast region, or Region 1. So I'm just going to be going over today some monitoring that we have planned for 2023. To start, we are continuing a lot of our routine monitoring. As Carly mentioned, Big Lagoon will be doing some monitoring. They are working with US EPA to do biweekly microsystems testing. They will also be participating in our partner monitoring project that I'll discuss here in a minute. Monitoring is going to be continuing in the Klamath Basin. This is largely the Hoopa, Karuk, and Yurok tribes, as well as Pacificor collecting samples to be analyzed by US EPA. This is also biweekly microsystems monitoring. And that image to the right there is just a map showing the various monitoring locations within the Klamath Basin. Tule Lake and Lower Klamath National Wildlife Refuge, the U.S. Fish and Wildlife Service was collecting some samples. It is my understanding that they may not be continuing due to funding limitations, but I can keep you all posted on that. Also in the Russian River, we have Sonoma Water conducting biweekly monitoring, doing visual assessments and cyanobacteria IDs. For free pre-holiday assessments, we have our water bodies listed here, and the holidays or dates in which they will be assessed. We also have some potentially more participants in the Klamath Basin, but I'll keep you all updated on that. So partner monitoring and special studies going on in the region. Like I mentioned, Big Lagoon is going to be collecting additional samples to their microsystems monitoring. They will be collecting samples for the 4 cyanotoxin suite analysis, as well as cyanobacteria ID. And this is going to be occurring, or actually started in mid-April and will be going through October. And then for special studies, there may be some additional monitoring in the rivers, including Eel, South Fork Eel, Russian, and Navarro rivers. Other studies and research, we are partnering with University of Nevada, Reno and CSU San Marcos, and they will be conducting some additional sampling to understand the spatial extent of benthic cyanobacteria and cyanotoxins, and this will occur in the Salmon and South Fork Eel rivers. We will also be working with US EPA on a project to develop standardized benthic sampling protocols. We will be participating in a training and one sampling event this season, and then additional sample events in 2024. And this is occurring in our region in the South Fork Eel River. And that's all I have. And just as always, would like to give a shout out to all our partner agencies. A lot of what we do would not be possible without their help. So thank you.
Great, thank you, Mike. Appreciate that. Do we have anyone from Region Board 2? I think they mentioned they had a conflict. Okay. Hal, you want to talk about East Bay Parks? Looks like you're on mute still.
There we go. Sorry, I had to use the phone situation because my computer's not working. Anyways, we have a fan bloom at Del Valle starting up. We're at a caution there. And at Quarry Lakes, at the end of March, we went to a danger. And last week we downgraded to a caution. That's a Phamazonon and Dolichospermum type bloom. We've taken down all our signage at Lake Temescal, and at Lake Anza we're having a benthic oscillatoria bloom. We're still at a no advisory posted. We'll probably be going to a caution soon there. And we're at a caution with our Shadow Cliffs dog area. We are in our weekly testing, and yeah, we're bracing for a big year. Anyways, that's all I have.
Thanks. And can you clarify, is the mitigation system still ongoing at Temescal? Is that one that was active this year?
We have one at Lake Anza.
Okay.
And we're planning on doing some Foslock over at Lake Temescal this year, but we're not planning on putting in an aeration system.
Okay. Thank you. Appreciate that.
Mm-hmm.
I know Sarah Ryan couldn't make it. I don't know if Curly or Marissa, I don't have the incident map up, but I think in general it's still been low for Clear Lake as far as recreational monitoring.
Aren't you about region 3 or region?
Clear Lake is 5.
Oh, 5.
But I know
okay,
I know Sarah usually covers it, but she's couldn't make it.
Yeah, the last data we had it still as clear.
Good.
So it's looking pretty good.
Good. I know Brianna Sakata is on. Do you want to do any report out for DWR?
Hi, sure. So we're just getting started with our state water project cyanotoxin monitoring. We began this week sampling at our water quality stations. And if no toxins are detected in the samples we collected this week, we won't sample again until the week before Memorial Day. And at that point, we'll be also including recreation areas in our samples, such as swim beaches and boat launches. And then our sampling at recreation areas will continue weekly until Labor Day. Our water quality site sampling is twice monthly at most of our sites, unless toxins are detected, and then we increase it to weekly. So our season's just getting started, but we did have one lake that had a bloom in our off season. That was Lake Paris, which is Santa Ana region, I believe. We had a persistent Planktothrix bloom that was present from December just until a couple of weeks ago. It did produce microcystins that were occasionally at the danger advisory level. The highest concentration in one of our samples was 326 micrograms per liter. But the bloom has finally subsided. The lake's under a caution advisory right now. The samples collected last week were all below the caution advisory level for microcystins. That's all I have for my update.
Great, thank you. And Corolla, I guess you're on. Do you have anything you want to mention for Clear Lake? Sorry about that.
No, sorry, I've been disconnected from that cyanotoxin monitoring program. I'm in the process of creating our fish program and the carp management. Matt, sorry.
No problem. Thanks for the clarification.
Just tapping in to keep up.
That. Yeah, no worries. I know Ellen and Janice are on. I don't know if they want to talk anything about from their Delta work. Just as far as a quick update.
Okay. So we finished a concentrated study at the Port of Stockton last year. A report is coming out soon. It's going to start by going to the port and city who supported work along with the freshwater harmful algal bloom program supporting that work. We don't have much planned for the Delta this year, except we saved money on the Prop 1 project, which was the cyanotoxins in benthic organisms, clams. So we will be able to continue some of that sampling this summer. And as far as Region 5 goes, we've started our partner monitoring program again with Restore the Delta. They will be monitoring weekly with test strips and then monthly toxin analysis sent to a lab in the Stockton waterfront and lower San Joaquin River. Carly and because we've got a vacancy, Carly and Marissa are helping us get, well, they're leading the other partner monitoring program with state parks. That's it. And we're hoping not to see anything until July.
Sounds good. Christine, Job, do you want to cover anything from IAP?
No, I don't have anything to report.
Okay, great. I know I had the workshop, which was great. Thank you. Anyone from Central Coast Regional Board or that area of the state? Any updates?
Yeah, Melissa's out, but they are partnering with their holiday assessments, and they'll be doing, I think, 5 or 6 sites this summer, adding onto their regular routine monitoring program. And then one thing to highlight is they actually did do an informational board item this month as to the FHAB program and how they're implementing it at their regional board. And it was received quite well. So that's their update.
Great. Any update from Region 4? LA area, either regional board or other partners? I know there was a report in of something on the coast, which is another one of these kind of overlapping areas between the programs that do marine hab and then what the water boards do for estuarine marine. So it's always interesting when we get to work through those overlapping coastal confluence areas.
That's right. We've gotten some reports from the public regarding some pretty dense algae blooms occurring in like the harbor around Orange County. And we're working with the local Environmental Health Department on investigating that, along with also the marine monitoring programs.
Great. Sabrina, anything for Region 6?
Good morning. Can you hear me? Yeah, I just have a pretty short update today. Since we've had such a large winter, and thankfully our season is off to a slow start. But we are planning to participate in the pre-holiday assessments again this season. Instead of sampling a lot of our water bodies for Memorial Day, though, we're going to do a post-holiday assessment. So we're going to do Independence Day, Labor Day, and then a post-holiday. We also typically see blooms later in the year in our region, so that is another reason why we're doing that. Also, like Carly explained earlier, we are expanding our partnership with Inyo County Environmental Health. So we are actually going to do a little bit more planning today. We're going to have a meeting, so we're going to figure out a little bit more which water bodies we're going to be monitoring. But that's exciting to have more monitoring in that area. We previously had some special studies at Red Lake and the Tahoe Keys, but this year, instead of continuing those special studies, we're going to be focusing more on some of the data analysis. We are still going to continue monitoring in those water bodies, but it won't be at the same extent. And then in the Tahoe Keys Property Owners Association does monitor on their own when visual indicators of HABs appear, so that will also continue. And they always share that monitoring data with us, so the HAB portal will stay up to date with all that information. And that's it. And I guess I did want to just say thank you to any of our partners, if any of you are on the call. I think our region is really trying to expand our partnerships, and if anybody ever wants to partner, please reach out.
Great, thanks, Sabrina. I don't see that Jeff is on. Any updates from Colorado River, Marissa or Carly, that you're aware of?
No updates for this year.
Okay. I forget, is the Salton Sea or the Colorado River and like Havasu part of the special study this year?
I'm not sure if they're part of the holiday monitoring, but they're definitely not doing any special studies. I don't think that they were at a point to mobilize to get a plan together for this year.
Okay.
Yeah, for the Salton Sea, there will be holiday assessment monitoring 3 times this summer.
Great, thank you. Thanks for pitching for all these areas. Appreciate it. It looks like Barbara was on. Barbara, do you have anything for Riverside?
Yeah, just a brief update that we've been working with City of Lake Elsinore staff with their ongoing bloom and high cyanotoxin concentrations. Since August of 2022, the lake has been at danger or near danger concentrations of cyanotoxins. So we've been working with them and Marissa and Carly with messaging to the public and with recommendations for their sampling. And we'll continue to work with them about public noticing. We're also going to be doing an eDNA study, participating with the Swamp eDNA metabarcoding monitoring and analysis project. And we're going to be pairing our FHAB holiday sampling for the 2023 field season with our eDNA sampling. The results will be used to support the SWAMP program and exploring the possibilities of eDNA in the FHAB program. And data from the 2021-2022 FHAB special study we conducted in Lake Elsinore and Big Bear Lake. It's also being compiled for uploading that data into CEDN and subsequent analysis. And in addition to the special study, we're also going to be participating in the holiday assessment this year. And that data will also be uploaded to CEDN. And yeah, that's my update.
Great, thank you. I know Carrie sent me slides, but it looks like she is on. Carrie, if you want to share those.
Sure. Let's see.
Sorry, Jamie, can you make her co-host?
Unless Jamie wants to go ahead, and I know she has them too.
Just share them for you.
Okay, whatever's easier.
Thank you.
Appreciate it.
How's that?
Oh, perfect. Thank you. So I'm Carrie Kowalski with Region 9, the HABS coordinator, along with Debbie Woodward. Just a few quick slides. I have 2 bloom updates and then what we're going to be working on this year. So you can go ahead and advance. So one of the notable blooms we had last year was in September at Cedar Creek Falls, and it was one that we caught on the tail end of our holiday assessments actually, and went back later. But it's a very, very popular hiking spot. A lot of people bring dogs, so it was important that we did this. And it was a big microcystis bloom that ended up having microcystins at a danger level for quite a long time. So through December, and then we finally had a good set of rains, and we were able to go back out in February, and the microcystins were back down to non-detect, and they could depost that area. So next slide. The other notable bloom we've had this year was at Spring Pond, and it was actually notification we received from Viejas tribal government. And thankfully, when we went out to their pond, there were cyanobacteria present, but the toxin genes were not. But as the conditions get warmer, we'll have them keep an eye on it. But the great part about this notification was that it actually opened the lines of communication now for us with the tribe. And they said that they would love any and all support that we have to help them do sampling when we can, and just to help them develop their own monitoring program. And we provided some of the great durable signs that State Board had given to us for their future use. And then next slide is just 2023. So we will also be participating in the pre-holiday assessments, and we have 6 streams that are high recreational use on our list. And then Carly already gave a great introduction about it, but we have 2 mini special studies that we'll do this year. And one's a continuation of a high agricultural use watershed that's the Santa Margarita River, where we found toxins in spat, and now we're taking a deeper dive to see what that really meant, and looking for the mats this time. And then our other new study is for the high quality streams. We've seen cyanobacteria present, and we have some DNA information from past bioassessments. So again, it's just a deeper dive to see, are they producing toxins? Is it something that we need to keep an eye on? And that's it. Thank you.
Thanks, Carrie. Okay, I think we've gone north to south. Apologies if I missed anyone, but feel free to chime in now. Okay, great. I know Marissa asked for co-host, and I have an update from the mitigation subcommittee. Hugh Dalton couldn't make it. I know they just had a meeting, and sorry, I'm trying to pull up the email. And those notes are available for attendees through Katie Fong, who's also covering for Joe today. So thanks for all your work on that, Katie. Too many emails up. Hold on. Here we go. The mitigation subcommittee took a break over the winter. As mentioned, they had a meeting in mid-March. They generally have monthly meetings during that. And again, you can find information through contacting Katie Fong or Hugh Dalton. During the winter break, there was one request for information through Sierra Lakes County Water District that they reached out to. They've also had discussions, as was mentioned earlier, with East Bay Parks with Temescal and Lake Anza that Hal McLean mentioned earlier. They've also been involved with Pinto Lake and City of Watsonville. So they're providing support. So again, if you're aware of a non-public or a smaller private water body that isn't public being managed, this is sort of a resource that's available to help fill in the gaps there. And so again, feel free to reach out to the mitigation subcommittee for that. They also have a web page on the HABS portal for resources for mitigating HABS as well. So feel free to reach out that way or check out those web pages as well. So thanks. And if you have any questions, again, feel free to reach out to them or join the next mitigation subcommittee call. And so with that, Marissa, are you ready? Are you covering Benthic?
Yeah.
Okay.
Yeah.
And then we'll switch back to, I think Shannon had a brief update for illness work group.
Right, I'm sharing my screen. And all right. So this is Marissa Van Dyke of the State Water Board and the Freshwater and Estuarine Harmful Algal Bloom Program. I'm giving a brief update on behalf of Mike Thomas, who is one of the leads for the of this reconvened subcommittee. So the subcommittee is going to be focused on the benthic guidance that was adopted by CCHAB several years ago. We're going to be reviewing more recent data, research, and use that to reconsider perhaps some elements of the guidance as appropriate. So we've talked quite heavily about reconvening the benthic subcommittee at the last two CCHAB quarterly meetings, so I won't belabor that information. Though after the last meeting, we did send out a survey to recruit participants and identify, subcommittee roles. That survey went out in the winter season, so we got quite a few participants interested in participating in the meetings. So thank you for everyone who volunteered for that. And we also have some researchers that would like to partner in a way of reviewing materials and providing technical feedback. So that'll be wonderful as well. So the first meeting to kick off the reconvened benthic subcommittee meeting will be on May 2nd. So next week over Zoom, so no traveling is needed. And then we're going to be sharing quite a bit of literature and things to review, to consider over the summer season, so that we can all get on the same page of like the kind of the historical information to then begin with monthly meetings starting in October of this year and run through the spring. There's no plan yet on when any revisions would be presented to the CCHAB network, but there would be plenty of heads up if it's going to be presented to the CCHAB network next spring or the following year. And with that, are there any questions about the benthic subcommittee being reconvened?
So Marissa, if people still want to attend, is that still possible, and how would they get the details?
Yeah, you can message the survey was already closed. So if you want to message the Water Board's program, we can discuss your interest and what kind of role you'd like to play. But we're hoping to finalize everything for the next the meeting next week.
Great, thank you. Shannon, did you have any update for the illness work group? I know you thankfully covered the outreach materials earlier.
Sure, real quick. Is it okay if I share screen?
Yes. Jamie, can you make Shannon co-host?
You got it.
Okay, very good. Hopefully you can see this. So thanks, everybody. I know I made my announcement before we started recording, so I'll just do a quick repeat. The HAB Illness Interagency Work Group, which is comprised, of course, for most of you know, OEHA, California Department of Public Health, Water Board, and Fish and Wildlife. And so we did our yearly updates for both freshwater and marine illness pages. These also include, so right here you can see this is what our new front page looks like. If you click on this button here, that goes directly to the same information in Spanish. We also have our updated fact sheet, which includes the new map for the data through 2022. So we have 5 years of data now. Here we have our marine version, and same thing also in Spanish. And there is a click to our HAB illness tracking fact sheet right at the bottom. So those are great for outreach purposes if anyone would like to share those with their stakeholders and people who would be interested in that information. One thing I wanted to draw your attention to on the freshwater side was that we did go ahead and aggregate the data for 2018 through 2022. So the aggregated data is represented by these blue bars. So we've had a total of 139 cases, and it's broken down by year. So the green bars represent 2022. So those are of interest. We had a total of 30 cases this last year, and that was broken down by 20 for human, 6 for fish, 1 for wildlife, and 3 for dogs. So a little bit lower for some of these cases compared to previous years, including 2021 in particular, with a few more human cases this last year. So again, please feel free to share that information with anyone who might be interested. We also updated the maps. These color maps represent 2018 to 2021. And for people who are interested in 2022 alone, this is what the map of California looks like by county. And just one other takeaway, we see that obviously during the warmer months with more people engaging in recreation and frequenting water bodies, we do see an uptick in cases overall. However, cases can occur at any time during the year, meaning blooms can occur any time of the year. So it's always advisable to practice best water engagement and safety measures, which those resources are also available on this website as a one-stop shop. So please feel free to reach out to the CCHAB co-chairs, or you can directly reach out to any of us who sit on the HAB Illness Committee. You can contact us through this page. Thanks very much. Any other questions?
Hey, Shannon,
Sure.
Can you scroll down back to the county area?
Mhm.
I just want to point out one other thing that we had gotten asked for was the by county case numbers. And so right above the map, because there's 58 counties, we have a link to the table. So that is the cumulative number by county, and we've just split for human and animal. So I know there was requests for people of particular focus on a given county. So we are providing that now, and that is available on the updated freshwater page. So hopefully that helps address the questions that we've gotten. So thanks for that. And thank you, Shannon. Appreciate it.
Thanks, everybody.
Great. Appreciate everyone sticking with us. So again, appreciate everyone's time for going through this. I'm going to just share the agenda again. So yeah, we've made it through. It's always a little bit hard to know how fast the HAB monitoring updates are going to go, depending on the time of year and who's able to attend. So appreciate everyone sticking with us, and we'll be able to get you out a little bit early. So the recording will be available. I put the YouTube page up in there. It usually takes us maybe until next week to get that live, but you feel free to check that out. And again, recommend that everyone sign up for the listserv to get updated information for upcoming meetings and the agendas, and that'll be August 2nd. Again, similar time frame. And then we also have our on the meeting page, also have our October date on there as well. So again, look forward to speaking with you then. And again, if you're interested in if you're a state, local, or federal government representative, we'd be looking to fill the co-chair spot. So we'll get information out about how to nominate, but start thinking about that. So appreciate everyone's input on that. And Jamie, anything else that I've missed?
I think just if anybody, as usual, if there's, we're working on the August and October agendas. So if there's any specific topics that y'all want to hear about, please let us know. And we've been kind of the last couple of meetings doing like themed research talks. So yeah, we're happy for suggestions. But I think other than that, you covered it well. And again, thank you, Becky, for your service on this committee. And I just want to say, like, we're sad to see you go. But yeah, you've just been really great on seeing this committee going for a long time. So thanks again for your
yes. Join the long list of alumni, of which quite a few still attend, which is a great sign. So thank you. Appreciate everyone's feedback. We will stop recording, I think, now. And again, we will have that information be out shortly. Thank you all.